Okay, good morning, everybody. So before we talk about the real stuff, I wanted to clarify some things. I know some students asking about the matrix and abstract. Um, this is an example that actually available in Moodle, right? You guys know, and so matrix and abstract should have three components. The first one is the matrix, this kind of table, and then the references, right? And then abstract. Yes. Should be the full list, right? And you only need to select three or between 3 and 5 papers from the whole list, right? And present here. But the other question that students ask is because this is an example, and this table, I mean, this matrix is specific for that uh, papers. So you guys probably will have a different story or something like this. So I think you can also use the matrix. Sorry, not matrix. Let me see. The evidence worksheet. Use this format to address the questions. Specific question. I think the other thing said he likes this better. So I don't know. I mean, it's up to you guys. Either you follow the other example matrix, or they use the evidence worksheet as a template for the matrix part, right? So again, we only need to submit the abstract matrix abstract. Follow the example. And in terms of format, you can use either evidence worksheet for the matrix, and also you can use the matrix in the example as a template. Both and either way is fine, right? And other things about the email, usually I send the whole group, the whole class, the email. I know you guys have a question and want to ask me. You can just click reply. And this actually, for me, is very difficult to checking because all the email that you guys hit reply, then embed in the same email. So sometimes it's very difficult for me. So if you have not received my response yet, but it is likely I might miss it. So if you have a specific question you wanted to ask me, try to send a separate email, right? Try not to click reply. And okay, so that's all for, let's see. Yes?
Do the references have to be within the last 5 years, or can they be like 5, 6, 7, 8 years to do like the history though?
Good question. Yeah, I think if depend on the topics you select, right? So if you are able to find enough reference, from past 5 years, that should be perfect. But if not, I think it's fine. Go a little bit older years, like maybe last 10 years or so, right? And also, you guys have a question about, the review article, whether you allowed to use the review articles. I think it's okay, even for the, professional review articles. Some people also use the review articles, but not like, many. For example, you have 10 reference, and you can use 2 or 3 review articles. That's fine. Just like relative kind of proportion, right? Yeah.
Does only one of the partners need to upload the matrix and abstract to Moodle?
Right.
Okay.
Yeah, just per group. Submit it as a group, right? Okay, so now let's start talk about the fatty acids oxidation. I think we finished the fatty acid oxidation of the saturated fatty acids that we took like palmitate as an example, right? 16 carbon, and we know that beta oxidation goes through cycles. Each cycle release 2 carbon molecules as the acetyl-CoA, right? So palmitate, complete oxidation of palmitate will generate 8 acetyl-CoA molecules, and during the beta oxidation, we also know that the FADH and NADH are generated, and the reducing equivalents will eventually go to the electron transport chain for ATP production. So you probably wanted to understand, the whole process. Started with the activation of fatty acids in the cytosol, right? Fatty acids activate to fatty acyl-CoA, and the fatty acyl-CoA needs to be transported into mitochondria because all the sets of enzymes for beta oxidation are localized in the mitochondria. So fatty acyl-CoA needs to be transported into the mitochondria, and the key enzyme, do you guys remember which is the key enzyme that help transport the fatty acyl-CoA from cytosol to the mitochondria? CPT1, right? CPT1 is a key enzyme that control the whole fatty acids oxidation rate, right? So the more specifically, the CPT1 is the enzyme that helps transport the fatty acyl-CoA from cytosol to mitochondria. And once they finish the beta oxidation and you generate acetyl-CoA molecules, and acetyl-CoA eventually enter the TC cycle for complete oxidation and generate additional ATPs and reducing equivalents, right? So you try to understand the whole process, starting from the fatty acids molecules into the complete oxidation CO2 water, and how many ATPs are produced, and this will help you understand the different process, right, in the fatty acid oxidation. So now we talk about the oxidation of unsaturated fatty acids. So if fatty acids have the double bonds, and how this unsaturated fatty acids can be it's oxidized, right? So here we give a linoleic-CoA as the examples. So linoleic acid has 2 double bonds, right? And we know that the beta oxidation always start at the beta carbons. So first of all, the 3 cycles oxidation. First of all, we run the 3 cycles oxidation and remove the 6 carbons from carbon 2 to 8, right? And you're generating 3 acetyl-CoA molecules, because this all the single bonds, right? So enzymes able to cleave the single carbon bonds between the carbon 2 and carbon 8, and then remove the 6 carbons, generating 3 acetyl-CoA molecules. So now the molecules linoleic-CoA then becomes the dieno-CoA, right? Dieno-CoA molecules, and you see that the double bond is very close to the beta carbons, right? And double bonds is not able to be cleaved by enzymes. So what our body needs to do is to rearrange the double bonds and to make the single bonds available for beta oxidation, or for the cleavage at the beta carbon position, right? So the next step will be the rearrangement of the double bonds. So the enzyme for this rearrangement is called isomerase, right? So isomerase that catalyze the rearrangement of the double bonds. So you see the movement of double bonds from this, let me see here, 3 and 6, 3, 4, 5, 6, right? 3, 4, 5, 6. Yeah. So double bond is in this molecules, double bonds located between carbon 3 and 4, right? So after isomerization, that double bond changes from the 2 and 3 positions. So then you expose or make the single bond available between 4 and 5, right? So in this position, that the enzyme is able to actually to break down the carbons here. So then you go another round of beta oxidation, and to remove the carbons 4 and 5, and generating one acetyl-CoA molecule, all right, here. And you just do this, again, rearrangement of double bonds and make, available a single bond for enzyme cleavage, and then eventually you will, let me see the last step here. So, okay, so eventually you remove all the single bonds, the carbons, and generate acetyl-CoAs. But eventually you will still have double bonds left, but these double bonds are not able to be cleaved. So that means you have less acetyl-CoA generated compared to the saturated fatty acids, right? Because the double bonds were not able to remove, not able to cleave, generating acetyl-CoA. So again, the unsaturated fatty acids oxidation, you compare to saturated fatty acid oxidation, will generate less acetyl-CoA molecules. So you guys just know the general concept for the unsaturated fatty acid oxidation, because of the presence of double bonds. So you will need the additional step is so-called rearrangement of the double bonds, and that the enzyme that involve this process called isomerase, and you generate less acetyl-CoA molecules. Okay, so I think we finished the fatty acid oxidation. Then now we move on to the ketogenesis or ketone body biosynthesis, which also considering the oxidative process. So when does ketone body synthesis occur? In which physiologic conditions? We mentioned that, right? Fasting, yeah, fasting or starvation. And this fasting is like prolonged fasting or starvation. That means that if we don't eat for longer than 2 days, and then our body start to making ketone bodies, right? We try to get alternative energy fuels, and this sort of the one relatively kind of physiological condition, right? But ketogenesis can also occur in some pathological conditions. In that pathological condition, when our circulation fatty acid levels rise, that will also promote ketogenesis, right? So this usually occurred in the very long starvation, of course, and also diabetic condition, diabetes, type 2 diabetes particularly, or type 1, type 2 diabetes, right? The reason is because in the in type 1 or type 2 diabetic conditions, because insulin either deficiency in type 1 or insulin resistance, right? Insulin cannot function well. So the glucose cannot be uptake into the cell for the energy. So inside of cell, actually, the energy crisis because of the less glucose available for energy, right? So body then will have to utilize the alternative fuel, primarily the fatty acids, right? So the lipolysis will be increased and then release a lot of fatty acids into circulation. So circulating fatty acids level is also very high in the diabetic conditions. So this will also drive the ketogenesis, right? So remember, the diabetic patients always have kind of very special smell, right? So it's because of the keto, high levels of ketone bodies in the circulation. Okay, so the next question: where does ketogenesis take place? In which tissues? The liver, right? So the liver is the only tissue that synthesize ketone bodies, right? Remember, only the liver and the mitochondria. The ketone body is synthesized inside of a mitochondria. So here, sort of the whole process, how ketone body is synthesized. The acetyl-CoA is the substrate for ketogenesis. So acetyl-CoA, majority of the acetyl-CoA molecules are from fatty acid oxidation, right? Thinking about the molecules, fatty acid has a 6. If the palmitate has 16 carbon, oxidation of one palmitate can generate 8 acetyl-CoA, right? And glucose only generated 2 acetyl-CoA molecules. So the fatty acids oxidation will produce a large amount of acetyl-CoA, and also under the fasting conditions, because our body wants to spare glucose, right? The glucose usually do not, in most of the tissues, do not completely oxidize, and usually just go the partial glycolytic pathway and stop at the pyruvate or lactate, right? So under the fasting condition, you have a very small amount of acetyl-CoA actually from glucose oxidation. So again, the majority of acetyl-CoA for ketogenesis during fast conditions from fatty acid oxidation. So fatty acids can have 2 different paths that can be completely oxidized through the TCA cycle for ATP production in the lot of tissues, particularly in the muscle, right? And in the liver, most of the acetyl-CoA are utilized for ketone body synthesis, right? And ketone body, once it's synthesized in the liver and released into circulation and picked up by other tissues and utilized as energy in the other tissues, right? And I wanted to tell you that the liver makes ketone bodies but does not utilize ketone bodies, right? We will go through the reason why liver cannot utilize the ketone bodies later, right? Okay, so and ketone bodies, and there are actually 3 molecules are considered ketone bodies, but I tell you that beta hydroxybutyrate and acetyl acetate are the major forms of ketone bodies, or in other words, the beta hydroxybutyrate acetoacetate are present in the most abundant, right, in the circulation in the body. And acetone is the byproduct of the decomposition of acetoacetate, and acetones usually is excreted in the lung. So that is a ketone, that is acetone that makes the special smell, in the diabetic patients or the ketone acetosis conditions. So again, these are the two major forms of the particularly beta hydroxybutyrate are the major forms of the ketone bodies, right, are present in the very high levels in the circulation. And these three ketone bodies can be interconverted, right? Acetate can be converted to 3-carboxyl beta hydroxybutyrate or vice versa. And acetoacetate can spontaneously composite to acetone and excrete it in the lung, right? So you will not be able to detect the acetone in the circulation because very low amount present, but mostly excreted, right? Okay, here is the process of the ketone body synthesis, biosynthesis. So ketone body again is synthesized from acetyl-CoA molecules, right? So acetyl-CoA molecules are the precursors of ketone bodies. So we already know that acetyl-CoA is a precursors or starting molecules for fatty acid synthesis, right? Now, acetyl-CoA is also the precursors of the ketogenesis, right? The ketone body synthesis. So, and again, the acetyl-CoA mostly is generated from the fatty acid oxidation, beta oxidation during the fasting conditions. So this is the acetoacetate. This actually forms from the two acetyl-CoA molecules, and acetyl-CoA together with the one acetyl-CoA molecules, right, generating the 3-hydroxyl 3-methyl-glutarate-CoA, or we also just simply call HMG-CoA molecules, right? So HMG-CoA molecules is synthesized from 3 acetyl-CoA molecules, right? And the HMG-CoA then is cleaved, right, and break apart into the acetyl-acetate and acetyl-CoA molecules. And this is an enzyme called HMG-CoA lyase, right? So break down the HMG-CoA into the two molecules. So relieve the acetyl-CoA molecule again, and this can be recycled for the second HMG-CoA molecule synthesis for the next cycles, right? And acetyl-acetate can be converted to beta hydroxybutyrate, and this is an enzyme called dehydrogenase, right? It can, catalyze this conversion. So this sort of a simplified version of the synthesis of ketogenesis. So first of all, the two acetyl-CoA molecules is generated a acetyl-CoA, right? And by this enzyme called silase, and this acetyl-CoA is breakdown. And together with additional acetyl-CoA molecules, the third acetyl-CoA molecules, right, and forming and formed HMG-CoA. And this enzyme that catalyzes the formation of HMG-CoA is HMG-CoA synthase, right? And then the HMG-CoA is breakdown to the acetyl-CoA and acetoacetate, and by this enzyme, HMG-CoA lyse. So I don't think you will need to remember all the detailed each reaction, but just kind of general pictures, all right? So for example, the substrate, what is the substrate of ketogenesis or precursors? Ketogenesis, acetyl-CoA, and where does the acetyl-CoA molecule from, right, comes from for the ketogenesis, basically from fatty acids oxidation, right? And the ketone bodies is synthesized started with 3 acetyl-CoA molecules, right? Of course, it should be considered from the 2 acetyl-CoA molecules, because the third acetyl molecules can just go the cycling, right? So, and acetoacetate basically is the 4 carbon molecules. Any questions about this ketogenesis? Right. Yeah, again, the ketone body is synthesized in liver, but not utilized by the liver, right? And these 2 major ketone body molecules usually just simply diffused into the peripheral tissue that do not involve the transporters or stuff. So that's the reason why we need to synthesize the ketone bodies I mentioned before, because the brain cannot utilize fatty acids as energy fuel, because the large molecules of fatty acids cannot be pass through the get into the brain for the energy. So we need to one of the very important reasons why bodies synthesize ketone bodies is under physiological conditions is to make ketone bodies they're smaller, so again simply diffusion into the brain, and brain can utilize it for the energy, right? Okay, so this slide shows us the whole pictures of the formation, utilization, and excretion of ketone bodies. And again, the high levels of fatty acids in the circulation is the most important driver for the synthesis of the ketone bodies, right? So liver picks up fatty acids from the circulation and synthesize the ketone body, right? And then release into back into circulation and pick up by the other tissues and utilize as energy in the tissue cells, right? And acetone again just excreted, and the other 2 forms of ketone bodies can be utilized in the other tissues. So in the peripheral tissues, that ketone bodies be break down again to the acetyl-CoA. This is just a simplified version, right? Simplified process. So again, to acetyl-CoA and then enter TC cycle for oxidation and ATP production. Okay, so utilization of ketone bodies. Again, we're showing that here fatty acids again pick up by the liver and to generate, go to beta oxidation first and generate acetyl-CoA and acetyl-CoA as the substrate and enter the ketogenic pathways for synthesis of ketone bodies by acetoacetate, and 3-hydroxybutyrate, and these two ketone bodies release into circulation and pick up by other tissue. Here we use muscle as example, and how ketone bodies are utilized, for energy generation. So first of all, 3-hydroxybutyrate be converted back to acetoacetate, and acetoacetate is break down to first of all it's processed the CoA transferase that catalyzes the conversion of acetoacetate to acetoacetyl-CoA, right? And this process is involved in the succinyl-CoA and succinyl-CoA generated, and this is the part of remember is TCA cycles, right? So acetyl-CoA once it's made, and then break down to the acetyl-CoA by this silase, and acetyl-CoA then enter the TCA cycle for complete oxidation, right? So here is a key enzyme for ketone bodies utilization, which is a succinyl-CoA transferase. Succinyl-CoA transferase is a key enzyme that converts acetoacetate to acetyl-CoA, and this is a key reaction for utilization of ketone bodies. And this process also utilizes 1-ATP, right, for the reactions. So again, this is a key enzyme. So the reason why liver cannot utilize the ketone bodies because liver does not have a succinyl-CoA transferase, right? So this is a reason. Try to remember this, right? The succinyl-CoA transferase is not present in the liver, so liver only makes ketone bodies but cannot utilize ketone bodies. Right? And of course, the ketone body also utilize in the main tissue, organ, the brain, right? Utilize a lot of ketone bodies for energy. And in terms of energy yield, from if we go, the tissue goes more, the ketone body. Let me see here. Oh, here I'll just try to make a comparison for energy yield if we utilize the palmitate versus the ketone bodies. If we, not completely oxidize palmitate, but stop at here and then make ketone bodies and then oxidize ketone bodies. See how, the efficient energy production. So for palmitate oxidation, goes beta oxidation will generate 8 acetyl-CoA molecules, right? And if this 8 acetyl-CoA molecules all enter TC cycle, and for complete oxidation, we already calculated, right? Will generate 129 ATPs. And if the acetyl-CoA generate from fatty acid oxidation and not completely, oxidize directly, but be utilized for the ketone body generation, and then a ketone body utilize ketone body for energy production, you will have actually less ATP produced if go to this process, right? So that's I think that's okay. Just gives kind of sense, But the problem is, well, not the problem, but thing is the this two different options will not happen at the same time or in the same cells or tissues, right? Because, for example, in the liver, most of the acetyl-CoA produced from the fatty acid oxidation will go ketogenesis. Only a few number of the acetyl-CoA will go complete oxidation for energy, right? And if that in the muscle cells, and muscle cells, I mean, depend on the availability, the fatty acids or ketone bodies. So the muscle cell usually do not utilize do not synthesize ketone bodies, right? So muscle cells will have two options in terms of the energy fuels, either fatty acids, so muscle can complete oxidize the fatty acid, go this pathways, or will have ketone bodies, so go this pathway, right? And I think, I'm sorry, I think this slide may be kind of a little bit confused, but just give you kind of idea, the ketogenesis, if palmitate goes to the ketogenesis process, will produce less ATPs in the liver, I would say, right? Okay, so now let's talk about the regulation of ketogenesis. So three very important steps that control the ketogenesis, the rate of ketogenesis. First of all, is the levels of fatty acids in the circulation. We already mentioned, right, a couple of times that fatty acids level are the main driver for the ketogenesis. So under the fasting conditions, when we have a lot of fatty acids that release from adipose tissue, basically due to the increase, the catecholamine levels, increase glucagon levels, then can increase the lipolysis, right? Remember we talked about lipolysis during the fasting, so then break down, triglyceride and release a lot of fatty acids into the circulation. That raises high levels of fatty acids. So this is one of the important control or regulation of the ketogenesis. Again, high levels of fatty acids can stimulate the ketogenesis. And the second point or step that is very important, also can control the ketogenesis, is the CPT1 gateway and 2 beta oxidation. Remember, CPT1 is a key enzyme, right, and control the transporter of fatty acids into the mitochondria for beta oxidation. So if this enzyme activities increase, then you have more fatty acids are transported into the mitochondria, and you have more fatty acids available for beta oxidation, and that will produce more acetyl-CoA, right? So that you will increase, substrate for ketogenesis. So again, the CPT1 gateway that control fatty acid transporter into mitochondria and beta oxidation is the second key point that can regulate the ketogenesis rate. And lastly, the acetyl-CoA levels, which make a lot of sense, right? If we have more acetyl-CoA, then you should have more substrate for ketogenesis. So anything that can control acetyl-CoA levels can also regulate the ketogenesis, right? So again, the three steps that can regulate ketogenesis: the circulating fatty acids levels, for example, during fasting, or during the disease condition like diabetic conditions. And second is the enzyme CPT1 enzyme activity that control the transporter fatty acids and beta oxidation, and lastly the acetyl-CoA levels, right, in the cells. All right. I guess we'll take a 10 minutes break. Right? And then, Okay, now let's talk about the ketoacidosis. And you guys probably heard about this before, right? Um, so ketoacidosis is a pathological metabolic state, right? It's not a normal mean. So it's because of this condition, because of the extreme or uncontrolled ketogenesis. That means, you have a very high activity of ketogenesis under the pathological conditions, and generate very high levels of ketone bodies in certain situation or conditions. So most common and ketoacidosis is most common in uncontrolled diabetes or the prolonged alcoholism, right? So we divided this ketoacidosis into like two categories. One called diabetic ketoacidosis. Other one is alcoholic ketoacidosis, right? So both diabetic and alcoholic ketoacidosis have common, sort of the metabolic problems, right? Both conditions, the common problem is that glucose utilization is decreased, right? So this will drive the body to utilize more fatty acids as an alternative fuels, right? So under the diabetic condition or alcoholism, because of the body or not able to utilize glucose as energy, so it has to mobilize the fat for oxidation for energy. So that will generate a lot of acetyl-CoA levels increase and drive the ketone body synthesis, right? Yes. This is a different condition, but alcoholic, yes, it does actually have causing the fatty liver disease. But fatty liver disease, we're going to talk about later, is caused by the accumulation of triglyceride, right? So it's a different cause, but it also have a problem with ketogenesis. So alcoholism really causes a lot of metabolic problems in the liver. So for example, diabetic conditions, why diabetic patients not able to utilize glucose, we already know the reason, right? Type 1 is because they cannot secrete insulin, so cannot transport glucose efficiently into the cell for energy use, right? So this is the one reason. And for type 2 diabetic patient, because of insulin resistance, so insulin action is reduced, so insulin cannot efficiently transport the glucose into the cells, so causing the similar issues. So glucose cannot be utilized as energy, although you have a lot of glucose available, right? You have higher levels of glucose in the circulation, but cannot get into the cells because of lack of insulin or insulin actions. And we also talk about, right, insulin dependent GLU4, right? It's a major transporter. So the diabetic patient also have a problem, have a reduced GLU4 levels in the adipose tissue. And in terms of alcoholism, because the alcohol, ethanol actually is being reported to inhibit the gluconeogenesis, right? So you cannot make a glucose, particularly during the fasting. And usually, alcoholism always have kind of problem with like vomiting, so you, and also they don't eat a lot of food, right? They drink alcohol. So alcoholism and these conditions, you also have less glucose being either made through gluconeogenesis, or you eat less food, or you have a less dietary glucose carbohydrate available. So all this reason, again, similarly causing the glucose utilizations decrease. So under this condition, that our body started to mobilize the fat as alternative energies, right? So you stimulate lipolysis and increase the fatty acids release into circulation, and all these are the major drivers. Remember, we talk about the three main steps that control ketogenesis, right? So first, the fatty acids levels increase in diabetic patients, and also alcoholism, in alcoholic, and then acetyl-CoA levels will be also increased. Like you generate a lot of acetyl-CoA from fatty acid oxidation. So all this will stimulate the ketogenesis. So this sort of the metabolic pathways, how ketone acidosis occurs in the diabetic patient conditions or alcoholism. Now let's go back, talk to regulation of the long-chain fatty acid oxidation, right? So this we just put all the lipid metabolism, lipogenesis, and fatty acids oxidation all together. See how under the different physiological condition, fat or fasting condition, that this lipid metabolic pathways are regulated, right? So let's talk about, so I highlight the red represents the pathway that under the fat condition, and blue under the fasting condition, right? So let's look at the fat condition first. So under the fat condition, we have a higher levels of glucose, right? So glucose can be oxidized under fat condition, right? So glucose is able to compete oxidize, right? So it's able to generate acetyl-CoA. And acetyl-CoA under the fat condition, that's generated from glucose, of course also maybe proteins, right? But not much fatty acids under fat condition, right? We don't need to utilize fatty acids because we have enough glucose carbohydrate. So acetyl-CoA basically is on fat condition, and mostly from glucose oxidation. And this acetyl-CoA will utilize for lipogenesis in the fat condition again, right? Particularly under the high carbohydrate diet. So lipogenesis will be stimulated. So, and this is the first key reaction for lipogenesis. Remember, the formation of malonyl-CoA from acetyl-CoA is lipogenesis, and this is a key enzyme. The acetyl-CoA carboxylase, ACC, is an enzyme, key enzyme. So this key enzyme can be regulated by hormones, right? So insulin stimulates and glucagon inhibits, right? Remember. So under the fat condition, because we have a higher insulin levels, so as ACC activity is stimulated, it's activated by insulin, and so that will leading to a generation of malonyl-CoA. So we have a lot of malonyl-CoA is produced from acetyl-CoA by ACC, right? So, and keep in mind this very important molecule, intermediate molecule again, malonyl-CoA. So malonyl-CoA is the very important substrate that can inhibit CPT1, Okay, so this here is the mitochondria, right? Mitochondrial membrane, and CPT1 is again localized in mitochondrial membrane and the key enzyme that control fatty acids transported, right? So under the fat condition, because we have a lot of melanoid-CoA is generated, so then CPT1 activities is inhibited by high levels of melanoid-CoA. So under the fat condition, we don't have a higher levels of CPT1, right? We have a lower CPT1 activity under the fat condition, so we don't have a much fatty acids being transported into mitochondria for oxidation. Does that make sense? So in the fat condition, right? So again, high glucose level and high insulin levels and stimulate lipogenesis, and you have a higher melanoid-CoA levels, and this will inhibit the CPT1, and you have not much fatty acids oxidation activity under the fat condition. So you have a lower fatty acid oxidation activity under the fat condition. Beta oxidation is lower. And you also have a lower ketogenesis, right? So we pretty much not able to detect the ketone bodies in the circulation under fat conditions, right? Because we don't need it, right? Okay, so now let's look at the fasting conditions. So under the fasting conditions, because of lower glucose, you have a low insulin, but high epinephrine and glucagon levels, right? So that particularly because of high levels of glucagon and epinephrine, that drives the lipolysis in the adipose tissues to break down triglyceride, right? Release a lot of fatty acids into the circulation. So we can see the fatty acids level in the circulation is up, is increased under the fat condition. So then we have a lot of acyl-CoA levels increase. And remember, this long-chain acyl-CoA is the important substrate that can inhibit lipolysis, right? Lipogenesis. Remember, when we explain the reason why high-fat diet suppress the lipogenesis, and this is molecules that long-chain acyl-CoA, or sometimes we just call acyl-CoA. Long-chain acyl-CoA or acyl-CoA levels increase because of high levels of fatty acids. And this is a key, again, substrate that can inhibit ACC, key enzyme in lipogenesis, right? So you can see the ACC levels is decreased, and we also another important regulators of ACC is the glucagon. So under the fasting condition, we have higher long-chain acyl-CoA levels and higher glucagon levels, and these two factors that drive inhibition of ACC. So we have a lower ACC levels and lower lipogenesis level under the fasting condition, right? So because of lower ACC levels, so we have a lower melanoid-CoA levels. And this inhibition will be gone, right? If we don't have much melanoid-CoA, then CPT1 will not be inhibited, so that under fasting condition, the CPT1 activity will be high increased. So this is again the key enzyme that increase then can transport a lot of fatty acids into the mitochondria and for beta oxidation, right, and generate a lot of acetyl-CoA, and that will lead to production of ketone bodies, right? So this, I think this is a very nice diagram to put all this, the regulation of the lipids metabolism under fat and fasting condition together. So try to remember the key things here, right? ACC definitely is the key enzymes, and marino-CoA is also the key substrate, and CPT1 is the key enzyme as well, right? So, and then see how hormones change in the two different physiological conditions, and how the substrate levels are changing, and and control the lipid metabolism. Any questions so far about the ketogenesis? Oh, I remember the student, she's not here today. She asking whether the low carb diet also promote ketogenesis. The answer is yes. I think the explanation is similar to the one of the slide that we because the low carbohydrate diet, we have a less glucose available, right? So the similar thing, you promote the fatty acids mobilization and oxidation, so that promotes ketogenesis. So there are also, if you guys probably also noticed that the diet called a ketogenic diet, Atkins diet, right? The ketogenic diet. Basically, they just no carb, zero carb, and you can eat, mostly protein and meat and vegetables, but zero carb. So this ketogenic, called the ketogenic diet, have a similar mechanism to promote the ketogenesis, because you have zero glucose available, and body will promote the fatty acids oxidation utilization. Okay, so I just wanted to summarize the and that we have covered so far for ketogenesis. So ketone bodies again are made in the liver, right, from the acetyl-CoA. So acetyl-CoA is the precursors of ketogenesis, and again the acetyl-CoA mostly from fatty acids oxidation, right, under the fasting condition, or even under the pathological condition. And mitochondria is the site of beta oxidation and ketogenesis, right? Both beta oxidation, fatty acids, and ketogenesis all take place in the mitochondria. And drivers of ketone body synthesis is increased fatty acids levels, right? Either during the pre-long fasting or under the pathologic condition like diabetic conditions or diabetes or alcoholism. And liver makes its ketone bodies but cannot utilize ketone bodies. The reason, you guys know, right, is because liver lacks of the key enzyme called succinyl-CoA transferase. This is a key enzyme that for the ketone body utilization. And ketone bodies, you just utilized by the peripheral tissue, and because of the molecules are small enough, so can just simply diffuse into the cells and for utilize energy. Yeah, I think that's all for Genesis. Maybe I'll just stop there, and then I'm going to go back to office to get the quiz, right? So you can have a couple minutes break, right?